Okay, good morning, everybody. Let's see if I can get this camera thing dealt with. I forgot. So we've got a handful of slides to wrap up this chapter. A lot of it, well, a chunk of it, I feel like, I hope, is some review as we think about things like osmosis and diffusion and how molecules might be able to move across a cell membrane or cell wall. When we think about bacteria, it will be a little bit different. Okay, that's better. So some of these things, as we get moving along, I'm going to go through a little more quickly. If you feel like you're a little shaky with that material, I recommend you go back and look through some of it. I'm not going to give you huge details of those ideas on the exam, maybe a little bit of stuff, all right? And we'll touch a little bit on that as we wrap stuff up today. So we have already shown some of these details. We've thought a little bit about bacteria and some of their morphologies. The next couple of slides go through and give you some perspective about this. And you will definitely see here shortly in the lab the opportunity to visualize some of them yourselves. So we talked about the coccus and the bacillus types of bacteria. We will see in the next slide some of their arrangements as well. So coccus or cocci, if we're talking a handful of them, are spherical in shape. We will talk about Gram reactions and all of that here in a second. We can see variations with all of these guys. All right. The bacillus are rod-shaped, and then we move into some differences. The vibrio are curved. They've kind of, depends on really some of them, but kind of comma-shaped as we look at them. And we'll go through more details of some of the pathogens a little bit later on. Coccobacillus is kind of a mixture of the two, an oval sort of shape. And then we move into the spirals, the spirillum and the spirochetes, corkscrew. Really, you know, these bottom two and our vibrio have some curvature, curved cells to that. And that is definitely a test question when it talks about curved and not, the bacillus is not curved, but the others are. Not our coccobacillus, but that's not one of the potentials. So here we can see some of the morphology or the arrangement, I should say. Often when we're looking at this, these are things we're going to be seeing under the microscope, right? And it might seem as if, how are you going to see them as groups of fours or groups of twos or whatever? And it's really, excuse me, it's the result of how these bacteria are going through their process of cell division, all right? And we're not going to go through great details with bacterial replication, all right? But we do think about in here the littlest bit of it, especially when it comes to this, all right? So as a bacteria divides and they replicate by what we call binary fission, they copy their nucleic acid, they get a little bigger and they split in half. It's really kind of similar to mitosis in our own cells. It's not exactly the same, but pretty stinking close, all right? So these cells, as they divide, potentially have specific ways or planes of division that they will divide into as they are replicating, all right? So here we can see just an individual one, all right? And sometimes as you're looking at a stain, you might see some cells that are all just kind of separate, whatever. And that's really nothing that's necessarily distinguishable or characteristic. It's just a bunch of cocci, right? There are a number of bacteria that as they divide, they basically split in half. So you get two of them linked together, diplococci, and you will oftentimes see lots of doublets all over the place, right? And that's just how they replicate, right? And after you've got those doublets, then they will go on and replicate and you get more doublets, all right? Is really what's happening, all right? I'm not gonna go through and give you examples of specific ones. We will see some of this later on, all right? That's not what we need to get bogged down with. The tetrads divide in two planes of division. Instead of basically splitting in half, we'll divide, it's two rounds of division, but we divide this way first and then we divide the next way and we get kind of stacked up groups of fours, all right? Technically, there's some more to it. We can think more three-dimensional and we can go, instead of 4, we could get 8 cells. We call that Sarcinia. That's, again, you know, for in here, we're like, well, you don't need to know that, right? It kind of looks like some dice as we've got the 4 little balls all hooked together is kind of what I imagine. And it can even get bigger as they start compounding, but we're not, again, getting bogged down with that. So groups of 4s, we call tetrads. And again, they're plane of division. The streptococcus can be very long chains, all right? There are some streptococci that are diplococcus, but a lot of the streptococcus genera have very long chains of cocci basically linked together. And as they divide, they basically start just kind of elongating. It's like the diplococcus, but instead of separating and keeping them in twos, we just keep it all connected, all right? The staphylococcus is more clustered, and instead of, you know, horizontal, vertical and horizontal planes of division to give us the diplo or the tetrad arrangement, this is all sorts of random, right? Different angles. And so we do get some little bits of chaining, but we also get all sorts of other angles that we're dividing, and we get bunches of cells, right? Clustered types of arrangements. And we call that arrangement Staphylococcus or Staphylococci, right? And a genus of bacteria makes these types of shapes as well. But not just because you see that doesn't necessarily mean that you know you're looking at Staph aureus, right? Staphylococcus aureus. There are other organisms that still are clustered, like Micrococcus. And so you can't just gram stain it up and be like, oh, that's a Staphylococcus, right? Morphologically, yes, that is how we describe it, but genus-wise, that's not, right? That lends to some confusion, right? When we start thinking about it. And the same with these chains, and we call it that, and there's also a genera that fit right within it, all right? The bacillus here, we can see them as single rods. There are also genera that are called bacillus, and they are shaped like this, right? Some of them, and really these up above with the cocci are definitely more characteristic when we think about seeing something under the stain and making some descriptions, right? Seeing them in tetrads or diplos or chains or clusters is important, all right? Seeing the bacillus, not that bacillus aren't important, all right? But seeing the bacillus, almost always, you're just gonna see a bunch of single cells, single rods, sometimes replicating, so they're 2 together. But that's pretty much with most of at least what we look at, that's how they behave. There are some that will link up kind of like our streptos up here in big long chains, and we call that streptobacilli. They're also described as cords or chains. But that is not something that is super diagnostic, all right? So usually, like I said, it's the staphylo or the coccus bacteria that we think about as describing more with the morphology, all right? You definitely might make a note of some bacillus that are in big log chains like that, because that is kind of a little bit different, right? But it's not something that you immediately are like, oh, I know it, right? So this next bit, we want to think a little bit about cells, and we'll show you a couple of other examples here. We're really more thinking just like a regular animal cell with a membrane. will, I think, show a bacterial cell and a plant cell because they have cell walls. They're kind of similar ideas as far as what's happening in a bacterial cell with a cell wall, all right? So this just gives us some ideas about these cells and their environments or their habitats. So osmotic pressure can cause issues with cells. It doesn't matter if we're talking our own cells or bacterial cells. And deep down, knowing the fact that dealing with different concentrations of things, this sets the stage for food preservation. As we think about things like making jam or salting out meats, you know, to preserve them, even drying them, making jerky and things like that. All of those make the whatever it is that we're trying to preserve super concentrated with specific solutes, salts, sugars, something like that, right? And that gets very inhibitory to organisms, especially things like bacteria. Sometimes mold is a little more tolerant with that, and you know, making some preserves, stuff like freezer jam and stuff, some of those things, maybe if it's not the best jam, that stays in the fridge for a bit, and you recognize after a couple of months that freezer jam's all moldy, right? You're like, good, because it sucked anyways, right? So kind of just depends, but bacteria wise, probably nothing's going to be able to tolerate super high concentrations of the sugars, right? So in an isotonic solution, in this situation, we've got a balance of solutes inside and outside of the cell. It doesn't have to be the same stuff, but they're balanced. And so the net movement of water is zero. It moves a little bit, but there's no real directional movement, all right? And that's what the arrows go in both directions are implying. Yes, water's moving, but it's not moving one way or the other. It's just hanging out, all right? So concentration's the same. Over here in a hypertonic, you got a boatload of solutes on the outside versus the inside of the cell. And now we think about osmosis. We think about water wanting to try to balance this out. And in these situations, the water goes out of the cell into the surroundings to try to equilibrate. And typically, that's going to be the demise of that cell as it's getting all of the moisture pulled out of it. It causes it to basically dry up, to crenate, if we're talking about like one of our red cells in a situation like this, or other animal cell, it gets all shriveled and puckered up, all right, as we're seeing up here, all right, crenation. So this also, we could think about like, you know, the jam, as you're making that, putting boatloads of sugar in there, it creates a very concentrated solution. And if there are microbes in there, it sucks the moisture out of it, right? And you're good to go. The opposite over here, a hypotonic solution, would be like, an example could be like distilled water, something that has basically no dissolved particles or ions within it. It's the solutes we're thinking about, right? So now there's no solutes outside of this cell. The cell has lots of solutes inside of it. And so the water wants to go from the outside to the inside of the cell to try to give the inside more water. We're like, we need more water. That cell swells up and potentially will rupture. If we're talking an animal cell, like a blood cell, it would fill up and lyse. If we're thinking bacterial cells or plant cells for that matter, we shouldn't be thinking plants in here. This is microbiology. But plant cells and bacterial cells with their cell walls in a hypotonic situation, the cell wall helps to provide some structural protection of that cell and keeps the plasma membrane from just exploding, right? For the most part, all right? You definitely can have some situations with, you know, like bacteria that may not be as accepting of some of those circumstances. So this is showing, again, a plant cell, but cell wall, and we can kind of see what's happening. And you would expect, although there aren't, you know, vacuoles inside that are dealing with some of the water, bacterial cells kind of appearing similar as far as what's happening with this, right? And I'm not going to get bogged down with that because Again, we're not talking about plants. So here is an electron micrograph of our bacterium, a rod-shaped bacterium, and we're trying to show this green dashed line around here is the region where the nucleic acids, the chromosome, and that's a bunch of little N's in here, where we expect to see it, right? And it's kind of just like the whole stinking inside is what I think, all right? It's not exactly, all right? There's definitely more bits out in this kind of periphery, if you will, that looks a little more grainy, right? A little more dark in here. And this region is really a region that's basically full of ribosomes. And so we got all of the nucleic acid in this nucleoid sort of region, middle bit, and then the fuzzy bit on the outside are full of a bunch of ribosomes. And as we are copying messenger RNA, as we're building messenger RNA within the nucleoid region, we start reading it right out in the fuzzy bit, right? In the periphery is where all of these proteins are getting made. And that's why it's kind of hazy, right? Because there's a lot of business going on in this bit, right? Not that there isn't a lot of business happening in the nucleoid region. There's high levels of activity, all right? And we're not going to get bogged down with this right now, but bacteria and our own cells, we go through this process of transcription and translation differently, all right? We still make messenger RNA, we bring in the transfer RNAs and we slap in amino acids, right? But in a bacterium, that stuff is happening almost simultaneously, all right? From almost the second messenger RNA is getting made, we're building proteins. And in our bodies, that doesn't happen like that. We build the messenger RNA in the nucleus, it gets shipped out of the nucleus, it gets modified and then shipped out of the nucleus, and then we start building proteins. It's a two-step process. Really, it's a two-step process with both. But in bacteria, both of those steps can happen virtually simultaneously, all right? So that we will touch on later. But as you can see this, there's no reason that it can't just automatically start doing that. We're not housed inside of a membrane-bound region. We don't need to get out of that. And we also don't need to modify the message, all right? It's already just information, all right? And that's not, again, something we want to really think details about right now. So here we can see the ribosomes, and these again would be that fuzzy bit that we saw in the periphery, right, of that cell. These ribosomes are going to snag on to the messenger RNAs that are being generated. They exist as separate subunits, two different proteins. There's some, the proteins, so enzymes, that will sandwich around that messenger RNA once it's basically detected. As this happens, you get a complex. We're not showing the messenger RNA in there, but you get a ribosomal complex sandwiching around that. And in bacteria or our prokaryotes, because archaea, similar idea, the ribosomes are smaller than they are in eukaryotes, all right? 30 and 50S for the small and large subunits, and in eukaryotes, it's 40 and 60, all right? So a little bit smaller in bacteria. And I guess deep down that kind of makes sense because these guys are super small compared to eukaryotic cells to begin with, right? So maybe makes sense. So this is just showing some of the potential inclusion bodies, not the spores with our bacillus and clostridium, all right? But other bits that might potentially show up in a gram stain as kind of a little hollow region, but with endospore stain, it wouldn't show up. And then you would know these aren't, that's not what we're thinking, all right? It's something different. And there's a handful of different things, lipids and other things, maybe even some pigments or sulfur granules or iron that can lend to some differences in there. And you don't need to get bogged down with what do they have, right? There's just potentials that maybe a cell might have some inclusion bodies, right? The spores, as we see in this image, is something that's probably more of what we might be thinking about, all right? Some of the other bacteria, you know, as we think like the sulfur or the magnetosomes or something, those are environmental microbes, not something that we think about as pathogenic. So the process of sporulation here, we are seeing that we've got a stain over here, a spore stain that's not the best, but there are some green spores that are visible. It's a little better right on the monitor, but projecting it up there, it's kind of lost. Up above is the process of spore formation, all right? And remember back to last time, that was so far ago, right? Remember back to last time when we talked about Tyndall and the process of heating and cooling products repeatedly. We want to think about this the littlest bit. It's to really destroy those spore formers, right? So as we see up above, our DNA, it kind of acts as if it's going to replicate and form two new cells, but it doesn't, all right? So it replicates the DNA, but instead of, you know, elongating and preparing to divide into two new cells, it starts to package up this newly replicated nucleic acid. It starts to form membranes around it. It packages some other stuff besides just the nucleic acid. There's a handful of enzymes that get packaged up inside of this membrane as well. Things that are going to be needed once the habitat goes back to being suitable, right? You need more than just your nucleic acid. You need the ability to really start the process of living again, if you will. Maybe we should say waking up because they're not dead. So we start forming this membrane around it. It forms this spore as it basically keeps depositing more membrane material, basically creating a very thick, almost impenetrable shell around this nucleic acid, all right? So the cortex forms around it, giving it greater levels of protection, and then basically the cell starts to degrade. It opens up, and what you're left with is just this spore. The rest of it, the nucleic acid and all of the cell degrades, all right? And you're left with this spore that can basically withstand all sorts of very adverse conditions, right? And not until it's back into a hospitable situation will it go through the process of hatching or exfoliating. Doesn't really hatch. It's not an egg, right? But it's kind of like that, all right? And as it exfoliates, it basically releases and starts, you know, comes out of its shell, if you will, and starts replicating, right? Yeah.
Is it like immediate or is it to get out of the school?
To exfoliate. So once that, it's not immediate, right? It takes it a little bit of time. And it's the same idea, like if you put a bacterium from, you know, if you took it off of your plate and put it into a broth, or if you took it out of the broth and put it on the plate, those cells are there, but it's going to take them a little bit of time to kind of get a feel for their new surroundings and really start growing, right? It's the same for the spore. As it gets in there and now all of a sudden it's back to room temperature or body temperature or plenty of nutrients, whatever it is, it takes it a little bit of time for it to kind of receive all of these cues and be like, wait a minute, we should exfoliate, right? And so with that, we think of things like the autoclave or the pressure cooker, bringing temperatures well above boiling, because just simply cooking this stuff up, bringing it up to a boil, doesn't do anything to the spore, right? But getting it above boiling temperatures with pressure added is a temperature that's great enough to kill those spores. So 121 degrees Celsius, cooking hot is sufficient to destroy those spores, right? So we mentioned Tyndall, that stuff wasn't a process, right? We didn't have that opportunity. And so he would take this vegetable broth and he would heat it and cool it over the course of days to make this process go over and over and over. right? And in the process of some of these cells forming spores, you get some of them that aren't quite ready to do it, and the temperature kills them, right? They didn't have enough time to form a spore, but a few of them did. And then they exfoliate, they start growing back as a vegetative cell, and we turn the temperature back up and we kill a bunch of them, and a few of them form spores. After you've done that for a week, you've basically, you've annihilated them all. There's none left to be able to do that, right? So this is the idea with that. And this is a different spore former, one that, we don't think about at all. It's really Bacillus and Clostridium that are pathogens, and the ones we're concerned about in here. So here's a membrane, plasma membrane, bacterial membrane. It's similar if we were thinking about other cells, but there are some differences as well with some of the substances exposed or expressed on the outside of that membrane can be specific to the microbe, which in turn, when we flip it around and think about our body's immune response, we are doing surveillance, looking for bad guys, looking for some of these things that they're showing. So it would make sense that some of the stuff we show on our cells would be different than what they are showing on their cells. And that's how an immune response really can function, by recognizing our stuff from their stuff, and differentiating that is critical. So this should, I hope, be sort of familiar as we look at some of this stuff. There are numerous types of proteins, some that are on one side or the other, others that are transmembrane proteins that span both sides of that phospholipid bilayer, right? With this, the phosphate head regions are hydrophilic, water-loving. The fatty acid tails poking to the center are hydrophobic, water-hating. And this creates a situation where now we've got a membrane that helps to keep the outside out and the inside in, right? With that said, we need to be able to have some substances to move through that membrane, right? Some stuff can readily go through the phospholipid layer. Other things need helpers like these protein channels. And with that said, we can also be moving just through simple diffusion down a concentration gradient from high to low concentration, or we could be going against a concentration gradient. And we call it active transport, where we use energy to push things into or out of that cell against its concentration gradient, all right? So various processes in order for that to happen. And then we'll think about like bulk movement of stuff with like the endocytosis. And there's a handful of ways that we can further divide that. So this is a pretty simplified example of simple diffusion, again, just based off of concentration gradients. So our green guys might be something like sugars or some other molecule that could, in this case, readily move through this membrane, all right? And it depends on specific molecules as far as what can pass or not. We're not concerned about that right now, all right? That's not really what we're getting bogged down with. We just want to see what happens over the course of time. So it's time-lapse photography basically on one image. At the first bit, we've got a lot of stuff on the outside of the membrane and very little or nothing on this side. This is the inside of the cell. And so we want to diffuse from high to low concentration, in this case, going across that membrane. And over the course of time, who knows how long, depends on the specifics. Temperature can be a factor. Concentration also is definitely a factor, all right? Things, if you're at, optimal temperatures and very high concentration, diffusion will move pretty rapidly, all right? You get to a point where we would say we are at equilibrium, all right? If you took the time, and if I'm boring and rambling on, you've probably already taken the time to count all of these suckers and realized it's the same on both sides. That's equilibrium, all right? And just like that isotonic solution that we showed a couple of slides back, we could put some arrows on there, go in both directions, because these molecules still have the ability to move, but there's no directional movement. Here, the arrows are all going down, right? We're all coming into this, but once we've reached equilibrium, we're just hanging out. We can move around a little bit. It's all fine and good, all right? So diffusion, it's a simple process from high to low concentration, really until you equilibrate, and then not so much, right? Facilitated diffusion is still diffusion. It's still a simple process, doesn't take any energy. It's just the movement through a protein channel. These are substances that can't just readily go through the membrane, so they need a helper, right? And it's the same idea, from high to low concentration, moving in, all right? Just with a helper, all right? And with that said, these protein channels do have specificity. Although we're seeing some of these things, it's really only allowing the green guys to go through here. The red ones, they can get through, but it's going to be a different mechanism or a different protein channel, all right? It's not just like an open portal that lets whatever the heck is passing by, you can just get through there because you happened upon me, right? It's not like that, right? It's recognizing and allowing specific things. There are other mechanisms where, as I said, the active takes energy. So we're going to push things against a concentration gradient. And when we think about cells, it doesn't matter if we're talking prokaryotes or eukaryotes, concentration gradients become super important. And when we think about like energetics, trying to like think metabolism, establishing concentration gradients and charge imbalances across membranes is super important, all right? So here we can see a sodium-potassium pump, and same ideas we've been seeing. The top is outside, the bottom is inside of the cell. These mechanisms require energy because we're pushing things against concentration gradients, all right? So here, we're gonna push the sodiums, our little green guys, which aren't the same as what we were looking about, you know, a couple of slides back, but it's not the same green guys, right? We should use some different shapes. These green guys are getting shoved out into the extracellular space that has more sodiums, right? It takes ATP to link up to this. It's a similar channel, but a little different, right? And these are kind of gated, if you will. It can close at one end or at the other, right? And the energy from the ATP molecule is what causes this protein to shift, right? To open on one side or the other, all right? So once the sodiums link up inside of here, ATP fuses and causes it to basically expel out those contents. 3 sodiums get pushed out. In the process of doing this, while it's still open, a couple potassiums, 3 out, 2 in is basically what happens. And both sodium and potassium are positively charged, right? So we pump 3 positive charges out, we allow 2 positive charges back in, and all of a sudden, we now have a charge potential that we are maintaining across this membrane, and that's important, all right? So we won't get bogged down with details with that, but active transport pushes these things, all right? And when we start thinking about metabolism, as we're going through all of this, if we're talking eukaryotes, we'll look at the inside of the mitochondria and see what's happening. If we're looking at a prokaryote, we will look at the plasma membrane and the cell wall, and there's a space in between. It's similar idea as what's happening in a mitochondria. Mitochondria were once bacteria, right? So this process is very similar, right? And it's a charge potential that's being generated. It's actively pushing these hydrogens against their concentration gradient. So we want to think a little bit about our bacteria, the cell walls. There's positives and there's negatives. And you don't need to get super bogged down with this, but recognize there are definitely differences. And really with the positives, we're stacking these layers up. And with the gram negatives over here, we're not getting all of these cross-linked and stacked on top. It's just one sheet, right? And it's kind of hard to visualize because it looks like it's pretty much the same stuff, right? But some is the same and some is different, all right? They both have this NAG and NAM and they're cross-linked together, all right? Doesn't matter, they're both doing it. But we also have some of these pentapeptides and tetrapeptides, although there's tetrapeptides over here. It's the pentapeptides really that are different, that allow the multiple sheets to stack up onto a positive, and you don't get that with the negative. It's a single sheet, single layer, right? So, and that's, although it looks pretty much the same, there's more to it. And we'll see that here in just 1 second, all right? So gram positives, super thick, gram negatives, very thin, all right? They also, the gram negative has an outer membrane, which is something you don't see in a gram positive, all right? So both of them, we've got the cytoplasm, we got all the guts inside of there with the nucleoid and all of the ribosomes pumping out proteins. We've got a plasma membrane. There's lots of similarities with the plasma membranes, pretty much same ideas as we look at that, all right? This bit up here in blue, maybe would be nice if it was purple because purple is gram positive. This stuff, the teichoic acid, or no, the peptidoglycan, sorry, teichoic acid is right what I'm looking at. These are spanning from the membrane all the way through, kind of tethering and anchoring it in place. And then the peptidoglycan were those stacks that I showed you just a second ago. But here, sheet on top of sheet, stacked up, all right? And kind of held or tethered in place with teichoic acid. With the negatives, it's only a single layer, all right, of that peptidoglycan. There's space in between, right? And there's really a little bit of space in between there too. We're not showing it, right? Single layer of this, and then that outer membrane. It's lipopolysaccharides, so fat, sugar, most of that's on the top. This is plasma membrane right here, right? Similar to that, but poking up in red are these lipids. And these things with gram negatives can cause some issues, can cause reactions. The lipid A and O antigen can stimulate definitely a very strong immune response, right? And we'll hit that stuff much later on when we start thinking disease. But some of the instances of issues with the bacterium might be just its surface, right? It's just the fact that it's got these substances can be really a big factor in what's causing some of the symptoms in the first place. So you can see differences between the positives and negatives. We already talked about gram staining and the procedure to stain these cells. You want to think more about that when we think test stuff. What are the steps? What would happen if we had this or that, you know, color-wise? Details about this, not so much, right? Maybe outer membrane and not, right? Or teichoic acid and not, but all of this other stuff, no, you don't need to worry about that. This is our acid-fast bacteria, so they still have a super thick peptidoglycan wall, gram-positive-like, all right? But they also have this mycolic acid, a waxy outer bit on top of the peptidoglycan that basically is like a protective coating, really, on the outside. So if everything's all fine and good and this bacterium is nice and fresh, really it shouldn't pick up a gram reaction because all of the mycolic acid makes it pretty much impenetrable to those staining reagents. But if the culture's a little old, mycolic acid might break down a bit, and then these organisms might appear gram positive, all right? So that's kind of why we're saying gram positive acid-fast bacteria. Really, when I start thinking about these guys, like in pathogenic micro, when I'm talking about them in lab, I don't call them gram positives because really they shouldn't have a gram reaction. We just call them acid fast, all right? But it does make sense. They would, with that thick cell wall, they would have a potential to take up that stain were they to be old, right? That also sets the stage for some potential issues, right? If you're expecting it to not gram stain and now it does, well, I'm pretty sure you're not going to be calling it mycobacterium, right? You're not going to think you're looking at tuberculosis because you would be thinking tuberculosis shouldn't pick up a gram stain, right? So it depends. If you're getting a sputum specimen from this patient, you're not getting an old culture, right? This is something that's hacked up fresh out of the lungs, right? It's going to be acid fast, right? If it's been something in the incubator for, 2.5 months, and then we stain it up, that might be a different story, right? So that's fine. This is just a blow up of that outer membrane of our gram negative. So here we can see part of that phospholipid layer. And then really, this is what we're thinking about with this big chain of lipopolysaccharide tethering to that outside bit of the phospholipid layer, and then a big long chain of proteins, O antigen. Again, we're not going to get bogged down with that. This image, trying to quickly get through this. We got a handful of slides left, but we're so close. We mentioned some organisms, maybe a good chunk of the pathogens, having a capsule layer, so an outer glycocalyx or sugar coating. And here we can see a lot of that. This clear zone around these cells are the capsule layers. So this protective mechanism. This is Pseudomonas, is a type of bacteria that's really pretty good, pretty proficient at forming these capsules. And with that, you can sometimes tell you've got a capsule former just by looking at the way the colonies look when you grow them up on a petri dish. You will see a lot of different types of bacteria. You're going to see some that look really kind of dry sort of appearance for the colonies, and others that look really shiny, almost slimy. Some do look very slimy, all right? When you see the real shine, the glistening types of cultures, the stuff that looks like it's slimy, almost always you could or should assume these guys are producing a capsule layer. There's a lot of polysaccharide that's being generated here. And if you were to do one of those stains, you would see stuff like that. So we also showed before some of the pili. In this case, this is allowing for communication between the two bacteria, the exchange of genetic material. This is not like bacteria, it's like bacterial sex, but it's not, all right? It's not the exchange of chromosome, but plasmid material, extra chromosomal material, all right? These guys can impart differences that the other cell maybe didn't have, right? There's other little hair-like projections coming off of these guys that we can see pretty well. These are the fimbriae, little hair-like appendages, not for attaching and communication between cells like these bigger pili or pilus if we're just looking at one. So this, we're going to look at it briefly and then show you some images real quickly. The flagella of bacteria, really, there's a little bit of difference between gram positives and negatives. Same sort of an idea, but having a double membrane, it's just a little bit different, all right, as far as how these guys are tethered or anchored in place. You don't need to get bogged down with any of that business. What we want to think more about is this, the arrangement, and then how they actually move, all right? So it depends, all right? And as I mentioned a couple of lectures back, it's the bacilli-shaped organisms. A coccus, you're not going to see flagella on them, all right? So the flagella can be a single one. We call that monotrichous, just one coming off of an end. Amphitrichous is on both ends. Lopotrichous is a big tuft, a group of flagella on one end, all right? A little tuft right here, right? And paratrichus, these are all over the cell, right? All over, like crazy hair everywhere, all right? Which makes you wonder, how would they swim, right? And that is impressive. So bacteria, it doesn't matter if we're talking one flagellum or many flagella, when they are moving, we call it a run, and these guys all function together. And the flagella, let me back it up too, they're spinning around, all right? Like a propeller on a boat spinning around, all right? And we've got this motor in here allowing them to move, all right? If we think about eukaryotes and flagella, if we were showing a sperm cell up here, you would see something that looks kind of similar, all right? But it's not the same, all right? And the way that it's functioning is different. And there are microtubules in eukaryotic flagella that are doublets, all right? And they're around and two in the middle. It's a 9 and 2 arrangement, but tubes, right? Microtubules, proteins, and they're cross-linked and they move back and forth like this, those proteins, and it causes the flagella to whip back and forth, not spin, right? Different, right? Same idea to move, but how it does it is a little bit different, right? So here, these guys are spinning, and if they're spinning counterclockwise, we're in drive, all right? We're going in a forward motion, all right? And we say that's a run. When we're ready to change directions, and this is all really just random, all right? We flip into this tumble. We change gears, if you will. We start spinning clockwise, but when we spin clockwise, now we're not functioning together. Now everybody does their own thing and spins clockwise, and it causes the bacterium to roll, to tumble, all right? They move by chemotaxis. They sense or smell, if you will, some sort of substance of food stuff, and they want to move over toward it, all right? But just as I started moving directly there, that's not what a bacterium is going to do. I might start off going this way, and I'm like, I'm pretty sure I smelled something. Then I change, and I tumble, and I move this way, and I tumble, and then finally I start on that right trajectory. But I'm probably going to do a couple more tumbles, right? It's not just a straight shot, all right? Many turns, all right? But there's definitely, in the presence of some sort of stimulus, if there is a situation where it could sense maybe some foodstuffs or something like that, it would be more runs and fewer tumbles than if it's just hanging out in a flask, minding its own business. It's going to be running and tumbling and just all over the place, all right? Different, all right? As far as how it gets there and what it's doing. Okay, we are very close, so close. The last little bit of this, and I've got just 2 minutes before we're out of here, just shows us some eukaryotic structure, some details of this, and I showed you a little bit before, all right? The idea is to give you a couple of examples of different types, and again, we're thinking microscopic eukaryotic cells. There's all sorts of different ones, and I see these and I'm like, oh, I want to tell you lots of stories, but I won't, all right? So nucleus is often the first thing you see, very well defined, and we could stain it and see it. Green would be the membrane around it, and red would be the nucleic acids that are staining. So a very nice definition of that, and 2 cells side by side. This other green is some other protein in there, cytoskeleton. We went through and talked about some of these details with the endoplasmic reticula and the membranes, all of this stuff I know we've focused on. It's just trying to reiterate that as we move through. So recall all of this part of this endomembrane system, the nuclear membrane, the endoplasmic reticula, rough and smooth, and Golgi apparatus are all part of this. So too are the lysosomes. They're all membrane-bound organelles that we see within these cells. And similarly, as we're looking at a plant cell, there's some other features with them as well, but this is just trying to show the Golgi. So I think really Although there's a couple of slides left, I think for the most part, I have covered everything that really is the focus for the exam. This bit right here, I will tell you, there are some videos that show some of this stuff functioning. This looks like it's caught right off of that. These motor proteins will literally walk along this microtubule, bringing a vesicle from one region to the next. And it looks like a little dude just carrying this thing, right? Awesome, right? Crazy eukaryotes. All right. Lastly, before you get out of here, I do know there's some stuff about this. So endocytosis is bringing things in. And we can divide that into phagocytosis, the bulk eating, where plasma membrane surrounds and engulfs and pulls it in. Penocytosis is cellular drinking, but it's pulling in solutes, not just water, right? And then receptor mediated, there's some sort of protein on the surface of the membrane, a specific molecule links up to it and it causes it to invaginate and pull in, all right? Exocytosis, do I not have that on here? Would be pushing things out the other way, right? And that's the idea with that. Okay. Farewell, guys. Have a great weekend. The test opens in 10 minutes. Not that I expect you to take it in 10 minutes. It is timed for 2 hours. Just a little over 50 questions. There's 2 matching questions and the rest are multiple choice. If you got questions or concerns, shoot me an email. Thank you.